Chapter 9, Immunity in the Lymphatic System, and Chapter 10, Infectious Disease and Epidemiology. And here we're going to completely go over Chapter 9 and most but not all of the parts of Chapter 10. Here are the primary objectives for this module, so feel free to pause the video and read these. Section 9.1, How do we adapt to stress? I think it's kind of strange that your book begins Chapter 9 by talking about stress and the fight or flight response, as you would assume that this would be more closely related to the nervous system. However, the fight or flight response and stress or stressors do have some effect or relationship with the immune system. So I guess that's why the book is putting them together like it does. But anyway, a stressor is any factor that causes stress. And a pathogen is any agent that causes disease. Typically this agent is biological. Pathogens are also a type of stressor. We deal with these pathogens using the immune system, which is the body's defenses system that works to counteract and destroy the invading pathogens. And we actually have two types of immunity: innate or non-specific immunity and specific immunity. In innate immunity, there are two types of defenses. There are external defenses, such as the skin and mucous membranes like those in the nose and mouth. And then there are the internal defenses, which are the general pathogen fighting measures of the body, which basically means they don't target any specific type of pathogen. And these internal defenses include antimicrobial proteins, fever, inflammation, and phagocytes or phagocytes. Specific immunity works to eradicate specific invaders, so it targets specific types of pathogens. And this type of immunity involves the lymphocytes of the lymphatic system. And we're going to talk about both of these types of immunity more in the following slides. Here's a list of the external innate defenses along with their functions. And there's also this little anatomical map of where some of them can be found. And I'm not going to go into this much because we're going to cover most of these in the following slides, but I'm just going to look at a few of them that we don't really cover. So there's the lacrimal apparatus or lacrimal gland, which produces tears, and the tears produced here dilute and wash away irritating substances and microbes. The urine can serve as an innate defense because it washes microbes from the urethra. Also, defecation and vomiting help to expel microbes from the body, and this would likely be caused by food poisoning. We're going to probably not go over most of these chemical factors later on. We're going to cover sebum though, so I'll start with lysozyme. And lysozyme is an enzyme which acts as an antimicrobial substance and is found in sweat, tears, saliva, nasal secretions, and tissue fluids. And this enzyme essentially interferes with the cell membranes of microbes such that the cell membrane is breached or a hole is made in it, and this allows water to flow into the microbe and kill it. Then there's the gastric juice, and we've talked about this earlier. This destroys bacteria and most toxins in the stomach. And then finally we have vaginal secretions, which can act as innate defenses. And that's because they discourage bacterial growth by being slightly acidic. And this helps to flush out microbes from the vagina. And obviously these are only produced in females. Here are the internal innate defenses. And I'm not going to really look at this at all because we're going to look at each and every one of these, but just use it as a study aid for the test. Now we're going to go on a little detour and talk about general adaptation syndrome, or GAS, which helps us to overcome or deal with stress. And general adaptation syndrome is an innate, automatic, physiological response to stress, which is also referred to as the fight or flight response. Gas occurs in 3 phases. In the alarm phase, the sympathetic nervous system initiates responses and stimulates the adrenal glands. The adrenal glands then release adrenaline, and this provides an immediate jolt of energy, allowing the body to better confront or escape the stressor. And then we begin the resistance phase, which is the prolonged response in gas. And in this phase, the body concentrates on surviving the stressor rather than evading it. In this phase, the brain consumes immense amounts of glucose, lipids, and proteins are also broken down, and we begin to conserve sodium and water. And this phase lasts until the stress is removed, the lipid reserves are depleted, or complications arise from the altered body chemistry. And then finally, the last phase is the exhaustion phase, which can be terminal as the extreme physiological conditions we're experiencing are difficult to maintain, and this could lead to death due to organ failure and organ system shutdown. Here are the 3 stages of gas, and I should specify how gas can be initiated as far as internal pathogens and immunity go. So obviously, as far as fighting internal pathogens, we're not going to be able to flee from them, so we're going to have to fight them. But anyway, the sensory signal to begin gas is going to have to come from internal receptors within the body. And just compare this to what we would typically think of when we talk about the fight or flight response, where we use our special senses to gather the sensory information, which is then sent to the brain. But anyway, alarm phase is the first phase of gas, and in this phase, the brain alerts the sympathetic nervous system to stimulate the adrenal glands, and this causes the glands to release epinephrine or adrenaline. The adrenaline boosts blood pressure, heart rate, and respiration rate. And also, at the same time when the brain's stimulating the sympathetic nervous system, the brain initiates energy release, and both the release of adrenaline and this energy release are going to lead to the fight or flight response. So in the resistance phase, the sympathetic nervous system affects organs other than the adrenal gland, and these organs include the liver and the pancreas, and the adrenal gland, obviously, and the kidneys, which causes an alteration in ion balance so that we can conserve water. And then in this phase, the adrenal glands actually release glucocorticoids, which are hormones that stimulate the release of glucose, which you're going to need for energy. And anyway, the adrenal gland and the liver and pancreas are going to cause a mobilization of the glucose reserves into the blood so that we can use them for energy. And also the brain's going to need this energy source. And then finally, in the exhaustion phase, and we don't always get to the resistance or the exhaustion phase if we can escape the threat or eliminate the threat quick enough. So here, the sympathetic nervous system stimulates it causes the kidneys to fail, and some other organs might also fail. It would also lead to the adrenal glands shutting down. Now that the glucose stores are gone, this can lead to the starvation of the neurons because that's their primary energy source, and all of this can eventually lead to death. And obviously, kidney failure would lead to death. But anyway, moving on. So since we're on the topic of stress, we're going to talk about post-traumatic stress disorder, or PTSD, which is essentially stress which seems never ending. And in this disease, it's essentially just a prolonged resistance phase in gas, and this may be developed after exposure to severe stress, such as warfare or violent crimes, such as rape or homicide. The symptoms of this disorder include fear, heightened vigilance, panic reactions, inability to concentrate, and memory disorders. And I think they probably should have just said paranoia instead of heightened vigilance, but they did what they did. In this disorder, the amygdala, which is the center associated with emotion and fear, and the hypothalamus, which is the hemostasis center, are the most often affected areas of the brain. And the only treatment for this disorder include psychotherapy or psychoactive drugs. I guess I should say treatments, because there's two of them. Section 9.2, Skin and mucous membranes are the first line of defense. So the skin is our body's outer layer of simple or stratified epithelium, which are supported by connective tissue. And in terms of the immune system, this is often called our first line of defense against pathogens because it provides a physical barrier. Mucous membranes also serve as physical barriers against invasion, and these are found lining any cavity open to the exterior, including the mouth, digestive system, respiratory, urinary, and reproductive tracts. There are also other forms of innate immunity covered in the next section, and these include chemical deterrence and general anti-pathogen measures. Skin is the primary physical barrier. Skin is actually considered to be the largest organ of the body, though I think the liver might actually weigh more. The skin prevents drying out and entry of disease-causing microbes. It also contains sensory receptors that monitor touch, pressure, and temperature. The skin also helps the body regulate water content and temperature, and it's also the site of vitamin D production. There are actually 3 layers of skin, even though your book makes it sound as if there's only 2. But these 3 layers include the epidermis, the dermis, and the hypodermis. And the hypodermis was formerly called the subcutaneous layer, but that term has since been replaced with the term hypodermis because it's just more accurate this way. This diagram shows the various components of the skin, including the 3 layers, and it's actually kind of out of date, and I had crossed out subcutaneous layer and replaced it with the term hypodermis, just to be clear that we're not going to call it the subcutaneous layer anymore. And we're going to talk about most of these components of the skin in the following slides. The outermost layer of skin is the epidermis, and it's primarily composed of keratinized, stratified squamous epithelium. And these epithelial cells are also called keratinocytes because they make a substance or protein called keratin, which is water insoluble, which means it ensures that we keep water outside of the skin and that we keep the skin dry for the most part. The outermost layer of the epidermis is composed of dead keratinocytes, which within the skin are our first line of non-specific defense. The epidermis also contains melaninocytes, which make a chemical called melanin, or rather a protein. And it's this protein that determines how dark someone's skin is. Also within the epidermis are the Langerhans cells, which are a type of defense cell, and the Grandstein cells, which help to control the immune response. And don't worry about these last 2 types of cells. because I took this slide from the BMS 110 class just because I thought it does a better job of describing the epidermis than the book does. But we don't really need to worry about these last two types of cells because I just want you to mainly focus on the keratinocytes and the melanocytes. The epidermis is also the pigmented or colored layer of the skin. Skin color actually results from the brown pigment melanin, which is produced by the melanocytes in the deepest part of the epidermis. And everybody has melanocytes, but they actually produce melanin at different levels within an individual, which accounts for the different skin colors in humans. So obviously, people with melanocytes that produce a relatively large amount of melanin are going to have darker skin compared to people who have melanocytes which produce low amounts of melanin who are going to have light colored skin. And now a tan results from UV light stimulating production of a specific hormone, which in turn stimulates the melanocytes to produce more melanin. So essentially, a tan it just occurs when someone has made their melanocytes produce a higher level of, or higher amount of melanin. And here in this diagram, you can see where the melanocytes are, and they're just this bottom-most layer of cells within the epidermis. The dermis is the second or middle layer of skin, and it's composed of loose irregular connective tissue. There's also some elastin and collagen fibers within this layer, which help to anchor the cells in place. The dermis contains the blood vessels and nerve endings within the skin, and it also contains some of the accessory organs of the skin, such as oil or sebaceous glands and sweat glands. And hair follicles and nails are also found within this layer of skin, excuse me. And then finally there's the hypodermis, formerly called the subcutaneous layer, and this is the layer beneath the dermis. It's composed of specialized loose connective tissue, primarily adipose tissue, and this adipose or fat serves as an insulator, and this is how the skin helps to regulate body temperature because we prevent heat loss through the skin because of this fat. And it also serves as a cushion within the skin. The skin contains several different types of accessory organs which help to lubricate and protect it. And these accessory structures of the skin include the glands, hair, and nails. One of these glands, or types of glands, are called sebaceous glands, and these are oil glands found within the hair follicles. They secrete oil onto the hair shaft, helping to keep the hair and the surrounding skin moist or supple. And the hormones of puberty increase the output of these sebaceous glands, which leads to acne. And acne is a physical change in the skin because of a bacterial infection in these glands. And also, some individuals produce more oil or sebum from these glands than others do, and that's what's responsible for some people having a greasier hair than others. The skin plays a role in temperature control, and one of the ways it does this is through the actions of sweat glands, which help to maintain thermal homeostasis. And these glands are tubes from the surface of the skin, which end at the dermis. They open at a pore, and down within the dermis, these tubes are coiled into a knot. And we produce sweat in response to rising internal temperatures. During average activity, your sweat glands produce approximately a cup or 150 milliliters of fluid per day. Hair is another accessory organ in the skin, and it's formed from the division of specialized epidermal cells within the hair follicle, and these hair follicles are located in the dermis. And technically, all the cells within this hair shaft are dead keratinized cells, similar to the cells found in the epidermis. And as this growing hair shaft pushes up, the older dead cells move away from the blood supply, and that's why these cells are dead. Hair serves as an insulator as well as protection for the eyes, nostrils, and ear openings, and it also helps to prevent loss of heat from blood flowing beneath the scalp. And over here you can see a picture, or rather a microscopic image of the hair shaft and the epidermal cells surrounding it. And then in the corner is just a diagram of the structure of hair. So up at the top is the hair shaft, and this is surrounded by the hair follicle, which goes into the dermal root, and it has the dermal root shaft labeled. And then at the base of the hair, this is called a bulb, and this is the part of the hair which contains live melanocytes and also receives blood from blood vessels within the hair follicle. The last accessory organ of the skin that we're going to cover are the nails, which reinforce the fingers and toes. And nails are actually flattened sheets of keratinized cells that protect the ends of these digits. So once again, we have dead keratinized cells in the nails also. The lunula is located at the base of the nail bed, and this is a thick layer of specialized epithelial cells from which the nail originates. And then the cuticle is the layer of epidermis which covers the base of the nail. And both of these structures are clearly labeled in the diagram below, so you can see the nail root, the cuticle, the lunula, and the nail body, or the actual nail itself. And then you can see the epidermis and the dermis, which is the layer from which the nail grows. And then you can see the finger bone within this finger. In addition to the skin, we also have other innate physical barriers, and these are the mucous membranes. They line any cavity open to the exterior, so they line anywhere in the body that's not technically in the body, or rather it's just a tube that runs through the body. And it lines the mouth and digestive tract, the respiratory tract, the urinary tract, and the reproductive tract. And this mucus within these tracts retards the growth of pathogens. The mucus within these membranes is secreted by the epithelial cells of that membrane, and thus we constantly wash the membrane. Often larger volumes of fluid wash these membranes as well, and this would include the urine in the urinary tract, the vaginal secretions in the female reproductive tract, and saliva in the oral cavity. In addition to our body's physical barriers, we also have innate chemical barriers that help to defeat pathogens. And I realize that we covered most of these in the chart we went over, but I'm just going to go ahead and review them again. So first we have the sebum, which is secreted by the sebaceous glands, and this sebum forms a protective acidic film over the skin, which is hostile to many bacteria. Then we have lysozyme, which is found in perspiration, tears, and saliva, and it's a natural antibacterial chemical which interferes with the cell membranes of bacteria, leading to their destruction. And then we have the gastric juices within the stomach, which have a pH of 2, which is a very unfriendly environment for many pathogens because they can't survive at such a low pH. And in addition to these chemicals, our body also contains strains of harmless bacteria which create a hostile environment for invading microbes. And one example of these harmless bacterial strains is a lactobacillus, which is found in the vagina. And these bacteria help to lower the pH levels, which helps to kill off certain fungi and bacteria which may enter the vagina. Section 9.3, we have a second line of innate defenses. So in this section we're going to talk about the internal innate defenses, which include antimicrobial proteins, fever, inflammation, and phagocytes, or phagocytes, you can say it either way. The antimicrobial proteins produced by the body are part of the internal innate defenses. And first we're going to talk about the complement system. And in this system, a group of proteins are stacked in a specific order to create a complement of antimicrobial proteins. And this created a complement functions like an antibacterial missile. The invading cell is impaled or run through with the protein complex, which allows water to enter and destroy the bacterium's internal chemistry, which leads to the death of that bacterium. Another antimicrobial protein produced by the body is interferon, which is a hormone secreted to affect nearby cells, and it serves as a chemical warning to these cells, and it helps to keep the viral infection localized. Also, the keratinocytes of the epidermis and both macrophages and neutrophils secrete antimicrobial proteins or peptides, and these macrophages and neutrophils are just two different types of leukocytes, and we've covered them earlier. The diagram at left shows how the complement system works, and just remember that the complement system is only used against bacteria. But anyway, I'm not going to get too technical with this diagram. Just know that in the complement system, we stack up these various antimicrobial proteins to create an antimicrobial missile of sorts, which we then shoot at the bacteria, creating a hole in its plasma membrane. Water flows into this hole, which disrupts the internal mechanisms of the bacteria, leading to the death of that bacterium. Then at the right is a diagram of how interferon fights viral invasion. And remember that we use interferon most often against viruses. But anyway, in this diagram we have an infected and dying cell which has been infected by this virus. And then we have an infected cell which isn't quite to that point yet. But anyway, this infected cell releases interferon, which is picked up by a neighboring cell, which essentially makes that cell resistant to this viral invasion, or rather it's prepared to deal with this virus. Fever harms pathogens directly and indirectly. So a fever is a change in the body's average set temperature, which results in an elevation in basal body temperature above 37 degrees Celsius or 98.6 degrees Fahrenheit. And your body's thermostat is reset by these chemicals called pyrogens, not to be confused with the pyrogens in pyrotechnics. But anyway, these proteins reset the body's thermostat to a higher temperature. and this may harm the pathogen directly, or the increase in temperature may harm the pathogen directly because it won't necessarily be able to survive at a higher temperature. Fever aids defensive mechanisms by raising the metabolic rate, which allows enzymes and repair processes to work faster because enzymes or chemical reactions take place more quickly at a higher temperature. This allows the cells to move more quickly also, and allows specific immune cells to be mobilized more rapidly. And when we have a fever, the spleen begins to sequester iron due to the higher temperature, and iron is needed by many bacteria for reproduction, so this is a way to begin shutting down bacterial growth and reproduction. However, this higher metabolic rate requires more energy. That's why it's a good idea to eat well when you have a fever. So that old saying, feed a fever and starve a cold, is at least true in the sense of you need to feed a fever. Inflammation is essentially just localized fever. So we say that it's in situ, or in place, swelling, redness, heat, and pain. And inflammation is triggered when damaged or irritated cells release hormones or chemicals called prostaglandins, proteins, and potassium. Mast cells, which are a type of leukocyte, release histamines that encourage blood flow to the damaged area and promote fluid buildup. And the causes of inflammation include pathogen entry, tissue abrasion, chemical irritation, and extreme temperature. The benefits of inflammation include temporary tissue repair, the blockage of continued pathogen entry due to the swelling, a slowing of pathogen spreading, and quicker repair of the damaged tissue. And usually redness in the skin is indicative of inflammation in the skin, but it also indicates extra blood flow to the capillaries due to this inflammation. Phagocytes are eating cells which indiscriminately eat invading pathogens. So the word phagio or phago means to eat, and cyte means cell, so that's how we get the word phagocyte. And these cells wander through the various tissues of the body, engulfing and removing anything foreign. And there are two basic types of phagocytes. These are macrophages, which are small cells typically found in the nervous system. And then we have the macrophages, which are much larger, and they actively patrol the cells, or rather they move through the various tissues of the body, picking up anything foreign. And some of these are fixed or residential, as is the case with macrophages also. But what we're saying here is that we have some fixed and residential macrophages, and these are localized to one area of the body. Section 9.4, the lymphatic system and specific immunity are our third line of defense. So the specific immune system, or the specific immune response, is both acquired and specific, which means that each type of pathogen which enters the body triggers a slightly different reaction from the body's internal defenses, and the immune system must learn to identify each type of pathogen through experience or past exposure. And we're going to talk quite a bit about how we acquire specific immunity, and most of this section is devoted to explaining that process. Specific immunity is governed by the lymphatic system, and this system consists of the various tissues, vessels, and organs which produce, transport, and store cells which fight infection. The lymphatic system reaches most of the body, and this system is composed of the lymph, the lymphatic vessels, and several different lymphatic organs and tissues. And it touches most of the body, which allows it to carry out both transportation and homeostatic processes. And these organs of the lymphatic system include the tonsils, the spleen, the thymus, the lymph nodes, and the Peyer's patches of the small intestine. And this network of lymphatic vessels connect the various organs of the body and collect lymph from these tissues so that it can be then deposited into the bloodstream as needed. And the lymph within the lymphatic system is actually a clear fluid that is called interstitial fluid when it is found in the interstices between the cells. And it has a similar consistency and cellular components as blood plasma, other than the fact that it doesn't contain erythrocytes. And also within the lymphatic system are these structures, or it calls them organs, called the lymph nodes, which are small bean-shaped structures within the lymphatic system. So they're pretty much just nodules that pop up within the lymphatic vessels. And lymph nodes are responsible for returning excess fluid from the tissues to the bloodstream. They also absorb fats from the intestines and transport those to the bloodstream. And this is actually the reason why lymph is white, or appears to be white and not clear, because it's full of these lipids. Also, lymph nodes help to defend the body against specific invaders by releasing the various leukocytes involved with specific immunity. The lymphatic system is essentially a type of circulatory system which runs parallel to the cardiovascular system. The lymphatic system also contains capillaries and vessels similar to what are found in the cardiovascular system. These lymphatic capillaries are found throughout the body, and these capillaries are themselves white because they contain lymph, which is white due to the lipids it carries. And the lymphatic system is an open system in contrast to the cardiovascular system, which is closed. And this means that the lymphatic system has a defined starting and ending point. And this is because the vessels lead from the tissues to the bloodstream, but not in the opposite direction. So lymph only goes from tissues to the bloodstream. The lymphatic system also has no central pump. Instead, it relies on the contractions of muscles to move the lymph through the vessels. And the lymphatic vessels are similar to the veins, as they also have valves to prevent backflow. The lymphatic vessels transport the lymph to either the thoracic duct or the right duct, which are the ducts responsible for draining lymph from the organs. And I'm not sure exactly why there's such an uneven drainage of lymph by the right lymphatic duct as compared to the thoracic duct. And you can see in this map that the right lymphatic duct only really drains the organs and tissues of the right arm and the right portion of the torso and the head, whereas the thoracic duct drains both legs, the abdomen, the left side of the torso, and the left arm. And your book really doesn't explain why, but I guess it's structured how it needs to be. This diagram compares lymphatic flow to cardiovascular flow. So in the top left portion of this diagram, you can see the arteries, capillaries, and veins of the cardiovascular system. And as you can see, blood flows from the arteries to the capillaries in purple, to the veins, and then back up to the heart, and then the whole circuit is completed again. Whereas with the lymphatic vessel in the light blue, you can see that it can only take or rather remove lymph or interstitial fluid from the tissues and then transport it through the lymphatic vessel and then into the bloodstream. And it doesn't go in reverse, nor does it really, well, it doesn't circulate back at all. So in number 1, it's talking about the blood in the cardiovascular system. And it says blood pressure forces the fluid portion of the blood out of the capillaries such that we can bathe the tissues. Whereas in number 2, it says the excess fluid is then forced into the lymphatic capillaries from the tissues by fluid pressure and also osmotic pressure. And then number 3, we're not really going to worry a whole lot about what this is showing, but you can just see that lymph flows without being pumped, and these vessels contain valves that prevent backflow, and it's just indicating a valve in a lymphatic vessel here. So this slide ought to clear up what I said about the lymph nodes earlier. The lymph nodes are small encapsulated glands strategically located to filter large volumes of lymph. And there's really not much difference between the lymph nodes and the tonsils. The only major difference is that the lymph nodes are encapsulated, whereas the tonsils are not. And the lymph nodes are found in the groin, armpits, and neck. The mesenteric lymph nodes form a chain at the center of the abdominal cavity. And these nodes act as filtering stations for lymph. Lymph enters the node via many passages, but can only leave by 1 or 2 exits, which forces the lymph to flow through the nodules in one direction. The lymph nodes can tell us a great deal about the health of specific regions of the body, and this is because they enlarge due to localized or systemic infection, abscess formation, or malignancy. So essentially, the lymph node closest to the affected area is the lymph node that swells up, which allows us to locate which part of our body is experiencing a malignancy or some sort of infection. And many infections and diseases can cause lymph nodes to swell. These include mononucleosis or mono, German measles, tuberculosis, mumps, ear infections, tonsillitis, an abscessed tooth, gingivitis, dental cavities, HIV and other STDs, rheumatoid arthritis, leukemia, and lymphoma. So, in many instances of infection and disease, we can use the lymph nodes to locate the affected area, except for a few of these mentioned diseases like leukemia or lymphoma, which may not cause a swelling of a specifically located lymph node. So here's a picture of a lymph node, and as you can see, these essentially look just like a bean connected to the ingoing and outgoing lymphatic vessels. And it has the capsule of the lymph node labeled along with the reticular fiber, which is just what makes up the inside of this lymph node and forms the lumen. And then within this lumen, if you will, of the lymph node, we formed a central maze, which contains both the phagocytic or eating cells and the lymphocytes. And you can also see the valves within the efferent and afferent lymphatic vessels. So here at the right, you can see some of the cells that may be contained within the lymph node, such as a macrophage or phagocyte, a plasma cell, which is a type of immune cell responsible for producing these antibodies, and then memory cells, which are a type of cell responsible for allowing us to gain immunity to infective agents that we've been infected with in the past. And then there are some bacterial pathogens shown, which I suppose could be within the lymphatic vessel, but I think here we're mostly just showing how the cells that are found in the lymph node might fight a pathogen. Or we're just showing them fight the pathogen. Tonsils, malt, and Peyer's patches are unencapsulated lymphatic tissue. The tonsils function nearly the same way as the lymph nodes. Like I said, the only major difference is that they're unencapsulated. And we actually have 3 sets of tonsils. There's the pharyngeal tonsils in the nasopharynx. There's the palatine tonsils on either side of the pharyngeal opening. And there's the lingual tonsils found on the base of the tongue. And these tonsils are open to fluids that pass through the throat, such that pathogens may become trapped in these organs, and this would cause these tonsils to swell, which can be severe enough to almost shut off the throat. And at this point, the tonsil would likely need to be removed. And then there's malt, or mucosa-associated lymphatic tissue, and also the Peyer's patches, which are a form of malt. And anyway, all of these malt tissues are egg-shaped masses found in the lining of the small intestine, or rather, Peyer's patches are found in the small intestine. The other malt masses are found in the mouth, and they help filter fluid. The Peyer's patches help filter fluid absorbed from the interstitial lumen. I don't know why we're even mentioning malt here if we're just going to talk about Peyer's patches, but it's just something your book decided to do. The largest organ of the lymphatic system is the spleen. This is a fist-sized organ, which is a collection of lymphoid tissue, and it has an outer capsule which surrounds the red and white pulp. Now, the red pulp contains erythrocytes, which is what is responsible for making it red, and also macrophages. And the role of this red pulp is to purify the blood by removing bacteria and damaged and or exhausted red blood cells. The white pulp of the spleen contains lymphocytes and is the portion of the spleen involved in specific immunity. And you can actually live without your spleen even though your immunity or your immune system's functioning level may be reduced, or it will be reduced. Another thing about the spleen is, since it filters so much blood, that if it ruptures, it will kill you unless you receive immediate medical attention. The thymus is responsible for producing mature immune cells, specifically the T cells. And if you'll recall, T cells are a type of lymphocyte, which are agranular, which means they don't take up the stains like the granular leukocytes do. And these T cells, if I haven't mentioned it already, take part in the specific immune system by directly attacking pathogens. But anyway, the thymus gland is located in the thoracic cavity behind the sternum and draping over the upper portion of the heart. And I believe the last time I talked about the thymus, I might have said that it was above the clavicle, but it's actually more or less below it. Actually, the clavicle goes over the top portion of it, it looks like. But anyway, it's over the heart, and it's really just between the heart and clavicle. And this thymus gland is composed of two lobes held together by connective tissue, so it essentially looks like a bow tie above your heart. And like I said, the thymus gland produces mature functional T cells. The cortex of the thymus specifically is involved in training T cells to distinguish self or our body cells from the invading pathogens. And the thymus causes these T cells to mature through the production of thymic hormones, which stimulate their growth and maturation. The bone marrow also produces mature immune cells, and to be more specific, bone marrow is actually the production site, or rather the birthplace, of all blood cells, including the T cells, and all types of blood cells actually mature here, except for the T cells, which mature in the thymus. And we're talking about the red bone marrow, which is found in the center of virtually all bones in children, but is only found in the skull bones, sternum, ribs, clavicle, epiphysis of the femur, pelvic bones, and the vertebral column in adults. And the bone marrow increases blood stem cells, which go on to produce or divide into both erythrocytes and leukocytes. And the cells which are involved in specific immunity are a subset of these white blood cells, as are all other immune cells. Now that we've talked about the organs of the lymphatic system, we're going to go on and talk about the cells of specific immunity. And like the book says, specific immunity relies on a series of deadly cells that recognize and remember pathogens. Now the immune system, here what is a specific immunity, is composed of a set of blood cells collectively called lymphocytes, and these are all agranular cells, so they don't pick up the stain. The various subtypes of lymphocytes look alike, but actually have different functions. They do, however, share some common characteristics. They have the ability to distinguish self from non-self, which means they can tell the difference between your body cells and pathogens. If you somehow lose this ability, or if your lymphocytes somehow lose this ability, your immune cells will actually attack your body cells and you develop an autoimmune disease, which we'll talk about later. They all have specificity, meaning they each react to only one particular type of antigen or pathogen. They have the ability to remember certain pathogens and use this ability to react more quickly the second or subsequent times that the pathogen is encountered within the body. And the leukocytes incorporate, or rather the lymphocytes incorporate two methods for combating pathogens. There is cell-mediated immunity, which is the type of immunity that T cells are involved in, and this is where these specialized lymphocytes, the T cells, function directly in any pathogen attack. And then there's the antibody or humoral immunity, which is mediated by the B cells, which are just another type of lymphocyte. And these specialized B cells function indirectly by helping create disease-fighting compounds called antibodies. The immune system uses two kinds of assassins, and we're referring to the B cells and T cells here. Now the B cells get the B in their name from the fact that they were first discovered in the bursae of Fabricius in fowl, which is essentially just an organ like the thymus, but B cells mature within it. And we don't have this organ in humans as the B cells, mature in the bone marrow. And these B cells spend most of their time inside lymph nodes and interstitial fluid once they've matured. They're the cells responsible for producing antibodies which are specific to a particular pathogen, which means they're part of antibody-mediated immunity, like we said in the last slide. And the T in T cells comes from thymus, where they mature due to the thymic hormones. The T cells actually make up about a half of the circulating lymphocytes in the blood. These cells are responsible for stimulating B cells to actually produce antibodies, which are then used against the antigen. And like we said, these are part of cell-mediated immunity because they're involved in the direct destruction of antigens. All lymphocytes have receptors on their cell membranes which detect the exact antigen that fits the receptor. And they can mix and match these genes, which are used to create the receptor proteins, to develop a specific protein for that antigen. And each of these lymphocytes is specific to only one type of antigen, and that's what allows us to deal with so many types of invading pathogens. But anyway, at your right you can see a lymphocyte. It doesn't say what kind, attacking an antigen or disease-causing a particle or cell. So B cells are involved in antibody-mediated immunity, and here's how. Each B cell produces a different antibody that is directed toward a specific antigen or infectious agent or pathogen. These antibodies are proteins that remove antigens from the bloodstream, usually by causing them to agglutinate. And once these antigens have been agglutinated, they're then endosized by macrophages or other phagocytes and then destroyed. The B cell wears this antibody or marker on its surface, and the marker reacts with the specific antigen, which activates the B cell, leading to it creating more copies of itself or cloning itself. And this process is called clonal selection because the antigen selects which specific B cell will be cloned. And then there are actually two different populations of clones which are created: the plasma cells and the memory cells. The plasma cells are responsible for pumping out an arsenal of antibodies directed against that specific antigen or pathogen. And once the antigen is gone, the plasma cells undergo apoptosis and die because we no longer need them. So now that the plasma cells have all underwent apoptosis and died because the antigen threat has been destroyed, how do we ensure that if we're attacked by the same type of pathogen again, how do we ensure that we'll be prepared to fight that pathogen again and don't have to go through this whole process a second time? Well, this is because we have memory cells, which are a type of B cell responsible for creating a library of long-term immunity that we call the secondary immune response. And this secondary immune response reacts much more quickly to an antigen than the primary or first immune response. And these memory cells are actually viable for as long as 10 years and stand ready to go into action. However, if the time period of 10 years or so has passed, we'll no longer have immunity to the pathogen and we'll have to go through another primary immune response again. And like I said, this secondary immune response allows us to quickly produce antibodies so that we can destroy the pathogen before it can cause us any real harm. And vaccines and booster shots are attenuated pathogens, which means they contain extremely weak or dead pathogens. And these vaccine and booster shots are designed to resemble a harmful pathogen, but they can no longer cause disease. the body then responds to these attenuated pathogens as if they were still completely capable of making us sick, and it then clones the proper B cells and produces both plasma and memory cells. And the reason why we have vaccines and booster shots is that they allow us to fight the pathogens without the individual having to get sick. This diagram shows how B cell activation works. So at the top of this diagram we have these inactive B cells which have these specific receptors or markers expressed on their surfaces. And when these markers come in contact with the specific pathogen, they bind to it and activate the B cell. And over there to the right of that B cell is what is known as a helper T cell, which can also lead to activation of a B cell. And once these B cells are activated, they clone themselves, cloning either plasma cells or memory cells. And these plasma cells then secrete antibodies against that specific antigen which activated the original B cell. The memory cells are the long-lived cells which remain in the body for years, so that if the same antigen invades, it can quickly be destroyed before it causes us any harm. Antigens are more specific than your social security number, and like I said before, these are proteins secreted by plasma cells in response to antigen binding. And though antibodies are very specific, they all share the same general shape, which is a doubled Y-shaped protein with one heavy chain and one light chain polypeptide. They all also have a variable region, which consists of the upper tips of the heavy chain and the corresponding tips on the light chain. And it's this region which is responsible for identifying each antigen. They interact with the antigen and cause it to agglutinate so it can be picked up by a phagocyte. A large conglomeration of antigen and antibody marks that antigen for destruction. And we have polyclonal antibodies which are directed against one specific antigen, but they link to many different antigenic sites on that antigen. And these are produced by many different plasma cells, or a group of plasma cells. Each plasma cell produces a slightly different antibody that responds to a slightly different portion of the invading pathogen, which just allows them to have many antibodies bound to the antigen. And then there are monoclonal antibodies, which are formed from the clones of one single activated T cell, such that all the antibodies are essentially exactly alike. There are actually 5 classes of antibody, but before we go into them, I need you to look at the diagram at your right, which shows the heavy chains of the antibody in purple, the light chains in blue. It also shows the variable region of the antibody, which consists of the top half of the heavy chain and all of the light chain. And these chains are linked together with these orange bars, which represent disulfide bonds. So back to the classes of antibody. So IgG is the most common type of antibody. It occurs in the circulating blood, lymph, and extracellular fluid. It binds directly to the antigen and activates it almost immediately. The IgM type of antibody is the first immunoglobulin released, and it's the predominant immunoglobulin produced in infants. And I should say it's the first immunoglobulin released during one's lifetime. It causes infected or foreign cells to clump together when bound by the antibody, and this aids in the release of complement. The IgA type of antibody can be a monomer or a dimer, which means it consists of two subunits, or it can even be a larger molecule composed, again, of Y-shaped units. One form of IgA found in the saliva can bind to pathogens before they enter the bloodstream. Then we have IgD, which you don't really need to worry too much about, which is found on mature B cells. It binds the antigens that stimulate B cell activation. And then we have the IgE type of antibody, which is an immunoglobulin, which is another word for antibody, responsible for immediate allergic reactions, and it binds to the surfaces of basophils and mast cells. And mast cells are just basophils, which are no longer in the lymphatic system. And it causes the release of histamines and other chemicals implicated in allergic symptoms. So probably if you're going to remember any two of these, it would be best to remember IgG and IgE antibodies and don't really worry too much about the other three. Cell-mediated immunity involves two kinds of T cells. So unlike B cells, the T cells must have the antigen presented to them first before they're activated. And this antigen presenting is done by none other than the antigen presenting cells, or APCs, and these are usually macrophages. When an APC encounters an antigen, it phagocytoses it, or eats it, and then presents a portion of that antigen on its surface. The APCs present their ingested pathogen using a specific membrane protein complex. However, just like B cells, T cells carry receptors on their surfaces that will bind to specific portions of an antigen. The T cell receptors only recognize antigens presented on the surface of an APC. T cells that recognize the APC presented pathogen are then stimulated to differentiate into either the helper T cells or the cytotoxic T cells, and either of these kinds of cells will make copies of itself to go and fight the pathogen, and it will also produce memory cells for fighting any further invasions or future invasions. This diagram shows how T cells are activated. So at the left of the diagram we can see the APC, or antigen presenting cell, presenting its antigen to an inactive helper T cell. Once the T cell is activated, it clones more helper T cells which secrete cytokines, and these cytokines are chemicals which go on to activate the cytotoxic T cells, another type of T cell. And the activated helper T cell also clones memory helper T cells. And then at the right we have activation of cytotoxic T cells, which we'll go into more detail later. But anyway, in this case, an infected body cell presents the antigen to the inactive cytotoxic T cell. This activates the T cell, it then creates more clones of itself, and these cytotoxic T cells then attack the infected body cells, and the T cell also clones memory cytotoxic T cells, and we'll talk more about cytotoxic T cells in one of the following slides. Cell-mediated immunity includes two large populations of T cells, the helper T cells and the cytotoxic T cells. The helper T cells travel to the lymph nodes where they stimulate the matching B cell. They help to bring the antigen to this B cell so that it can begin making antibodies. And then there are the cytotoxic T cells, which are stimulated to divide by cytokines released from helper T cells. They respond to specific altered HLAs or human leukocyte antigen proteins. This HLA complex is a marker that identifies cells as belonging to the body. And these HLAs are what we use to identify tissue type in people so that we can properly match them to their organs in organ transplants. And the cytotoxic T cells remove any cell without the proper HLA antigen, whether it's cancerous, an infected body cell, or if it's a cell beneficial to the body, but it's just presenting the wrong HLA. And cytotoxic T cells physically attach to foreign HLA carrying cells and release a chemical called porphyrin from their vacuoles. Here's how porphyrins work. Porphyrin molecules are essentially molecular bullets which make holes in the plasma membrane of the infected cell. These holes then allow salts and water to enter that infected cell, which causes it to swell and burst and then die. So over here at your right, we have how this process works. So we have this activated cytotoxic T cell, which it binds to the infected body cell by way of this antigen that it's been presented with. It then releases porphyrin, which makes a bunch of holes in the plasma membrane of this cell, which then causes it to swell and burst, and it kills the cell. T cells can also differentiate into another type of immune cell, and this is the natural killer cell. Natural killer cells, or NK cells, are actually part of our innate immune system or defense system, which means that they kill pathogens unspecifically. However, they're produced exactly like the helper T cells of the specific immune defenses. These natural killer cells patrol the body and identify either virally infected cells or cancer cells. And they kill via cell-to-cell contact. They release porphyrin, which creates pores in the cell, which leads to its demise. They also release other proteins, which cause the infected or cancer cell to go into apoptosis when they take in these specific proteins. Section 9.5, immunity can be acquired either actively or passively. So there are two ways to develop immunity. You can develop it through active immunity, which is where your immune system is actually made to respond to an antigen at some time during your life. And here your immune system will need to actively combat and successfully destroy the pathogen. And then there's passive immunity, which occurs when antibodies are transferred to the body without having to stimulate the immune system. Active immunity is the trainable part of the immune system, and the primary advantage of this type of immunity comes from the creation of memory cells. So initially, when we're infected with a pathogen, our immune system carries out a primary response, which may indeed take days to respond to the pathogen. Here we must stimulate the proper cells and follow the chain through helper T cells to B cells, and then to plasma cells, and finally to antibody production by those plasma cells. The body then needs a few more days of antibody production to elevate the antibody levels to an effective level such that we can finally completely destroy the pathogen. These memory cells then remain in the body for years, lying dormant until the same antigen reappears, and then they can start the secondary response. And this secondary response happens much faster than the first response because the immune system needs only to stimulate and clone the memory B cells. However, the disadvantage to this type of immunity is that it requires previous exposure to the pathogen, the onset of illness, and a successful recovery, though the advantage that we can then take care of the pathogen quickly shouldn't be overlooked. One way to achieve active immunity is through the use of vaccines. And before we go too much into what the book says about vaccines, I was just going to say that the idea that vaccines lead to autism is a complete and total myth. There is absolutely no sound scientific proof that shows that vaccines actually lead to autism, no matter what celebrity may tell you that they cause autism. But anyway, back to the actual science. Traditional vaccines use an attenuated viral particle with an intact protein coat, but no capacity or capability of causing infection. And these attenuated viruses are injected into a healthy person such that they trigger the production of antibodies toward the viral coat, and they allow for the production of memory cells, the same as what occurs when we're naturally infected with a pathogen. And I'd also like to say that sometimes if you get a vaccine shot, you might get a little sick for a little while, but it's going to be nothing compared to actually being exposed to the healthy and completely functioning pathogen. This graph provides a comparison of the response times for primary and secondary responses. So in the primary response, you can see that it takes about 10 days to start producing any type of antibody. And also in the green line, where it shows the total antibody, we have a plateau, which basically indicates that it takes us a while to completely combat and then destroy the pathogen. And then once we've destroyed it, we go into this decline of the antibodies because we no longer need them. However, in the secondary response, we start producing antibodies at around 5 days, so already we're twice as fast. And then we spike up the total antibody production very quickly and quickly destroy the pathogen. And then our total antibodies within the body begins to decline. Passive immunity gets help from the outside. So in passive immunity, antibodies are transferred to an individual from another source, and this does not require stimulation of the immune system. Passive immunity is beneficial because you do not need to expend any energy creating antibodies or producing clones. The transferred antibodies provide the recipient with immediate resistance to a specific antigen. However, immune protection is lost once the antibodies are used up or broken down because we can only receive a finite amount of antibodies. And we can't make more because no memory cells were ever cloned. These are used to fight diseases that cannot be fought in any other way. And some examples are maternal antibodies, which are passed through the breast milk of the mother to the infant, or gamma globin shots, which are just a type of shot where antibodies are injected into an individual. So that's all we're going to say about active and passive immunity, because we're going to begin talking about some autoimmune diseases here briefly. In an autoimmune disease, defense becomes offense. So an autoimmune disease is when the immune system mistakenly attacks the body's own cells, tissues, and organs. So they're essentially the result of no longer being able to differentiate self from non-self, or rather the cells of the immune system can no longer distinguish our own body cells from pathogens and attack them. And there are several different types of autoimmune diseases, each of which has a different effect, and this effect depends on what tissue is under attack. So here are 3 examples. In multiple sclerosis, the attack is directed against the nervous tissue, and in multiple sclerosis, we actually have T cells attacking the SWAM cells, which produce melanin around the axons of the neurons, and they just destroy them to such a point that we can no longer send nerve impulses efficiently, which explains the symptoms of multiple sclerosis. However, the SWAM cells can repair themselves or repair the melanin sheaths from time to time, which explains why multiple sclerosis it comes and it goes, essentially. And then there's Crohn's disease, which is an attack directed against the absorptive portion of the gut, such that we can no longer absorb nutrients in the correct manner. And then there's type 1 diabetes mellitus, which is where the immune cells attack the pancreas. And the site of the attack can vary. One example of this is lupus, which may affect the skin and joints. And in another case of lupus, it may affect the skin, kidney, and lungs. And then there's rheumatoid arthritis, which is, interestingly, an autoimmune disease. And in this disease, the immune system attacks the joint capsules of the body, leading to very painful and deformed joints. Many times the damage caused by an autoimmune disease may be permanent. And one example of one that is permanent is in diabetes, where the insulin producing cells of the pancreas are permanently destroyed as they do not regenerate. And one interesting fact about autoimmune diseases is that though they afflict millions of Americans, typically more women are affected than men. And some autoimmune diseases are frequent in certain minority populations. For example, lupus is more common in African American and Hispanic women than it is in Caucasian women of European ancestry for whatever reason. It's just the difference in genetics, essentially. And then rheumatoid arthritis and scleroderma affect a higher percentage of some Native American communities than the rest of the general U.S. population. Moving on to Chapter 10, Section 10.1, the study of epidemics is global in scope. So here we're just going to define some of the terms out of Section 10. Outbreak is the sudden appearance of a disease in a small localized group of people. Compare this to an epidemic, where many individuals are affected at once, and the disease spreads from person to person in a geographic area where the disease is not typically found. And then a pandemic is where a very large number of people are affected over a very wide geographic area, such as the entire globe, for instance. So now we're going to jump to the part of Section 10 where it says the disease process has several stages. So first, the pathogen enters the host and alters the physiology of the body to ensure the survival of that pathogen. The entry of the pathogen requires a specific mode of transmission, such as physical contact with an infected person, contact with the airborne pathogenic particles, or direct injection of the pathogen into the body. And pathogens attack their host in several different ways. Bacteria adhere to host cells, colonize host tissues, or even inhibit the host's typical immune response, whereas viruses often invade and take over host cells. And this is, well, in viral infection, requires that host cells exhibit virulence factors, which are specific proteins that the viral particle can bind to, and these vary across species and explain why certain pathogens can cause illness in one species but not in another. So here is a list of some epidemic diseases along with the pathogen responsible for causing them, the symptoms, and the location. And we're not really going to look too far into this list, but this will be helpful in the following slides because we're going to go over these diseases, or rather over what sort of pathogens cause them, so it's helpful in that respect. So you don't really need to worry too much about the symptoms or the location even. Section 10.2, Bacteria are single-celled wonders that can cause disease. So the rest of chapter 10 is mostly concerned with the different types or classes of pathogens, and we're going to start with bacteria. It's estimated that there are between 2 and 3 billion bacterial species on Earth. However, less than 0.5% can cause disease in humans. And actually, many of the most troublesome diseases are viral, but there are still several bacterial diseases which can be difficult to treat. Each class of pathogen, and here we're just talking about the different classes, which are bacteria, viruses, fungi, protists, and prions. Each of these classes of pathogens has distinctly different characteristics, and each requires different treatments to overcome. So we've covered bacteria and prokaryotic cells a little bit in the previous modules, but here in this section we're going to go much more in depth, so only a small portion of this is actually review. But anyway, bacteria are prokaryotic cells which can be found in the ground, water, and air, and within the bodies of other organisms. And in fact, there are actually much more bacterial cells living within our body than our actual body cells that make up our body. And there are actually over 700 different species of bacteria which live in our large intestine. But anyway, these prokaryotic bacterial cells are smaller than eukaryotes, and they range in size from 100 nanometers to 7 microns. So here at your right, you can see in the picture that's the largest species of bacteria discovered so far. It's called Thiomargarita namibiensis, and this would be about 7 microns. And you can compare this to a typical human cell, so it's much bigger and much larger than the E. coli over there, which is just the dot. Anyway, bacteria have no internal membranes and no division of labor. So here we're essentially just saying they have no actual membrane-bound organelles, and they have no nucleus where DNA is stored. So their DNA is just kind of all lumped together in a region called the nucleoid, which has no actual membrane surrounding the DNA. Any special function that bacteria need to carry out, such as photosynthesis or cell respiration, must be carried out through the aid of the cell membrane. However, bacteria do have ribosomes like eukaryotes because they're going to need to translate protein from RNA as well. Bacteria are classified by shape, staining, and genetics. Bacteria can be either rod-shaped or spherical, and these round spherical bacteria are called cocci. Rod-shaped bacteria are called bacilli, And there's also a twisted rod shape called spirillum. And typically these words are put into the scientific name of bacterial species. Bacteria can either be gram-positive or gram-negative, and this is determined using a staining technique called gram staining. The gram-positive bacteria retain a purple color from the crystal violet dye used in gram staining, whereas gram-negative bacteria pick up the red dye stain sapharinin. And which type of dye bacteria pick up is dependent upon the qualities or the thickness of their cell wall, and that's as much as I'm going to get into gram staining. Bacteria can also be classified their genetics, and we do this using a technique called DNA-DNA hybridization, and this allows us to compare the DNA of two types of bacterial strains to see how or to what degree their relatedness is. And I'm not going to get too far into DNA hybridization, just know what it's used for. Humans actually live with more than 2,000 types or species of bacteria within their bodies. And like I said, there are many more bacterial cells living in our bodies than there are actual body cells making up our body. And in fact, it's estimated that if you have 10 quadrillion cells in your body, you may have as many as 100 quadrillion bacterial cells living within your body. So this drawing just shows the difference between the two basic bacterial shapes. So at your left we have a cocci-shaped bacteria compared to a bacilli-shaped bacteria. And as you can see, they're made up of the same structures and only their shapes differ. And below that we have some pictures of cocci and bacillus bacteria. So we have these cocci bacteria, and over to the right of those are streptococci and staphylococci, both of which can cause either strep or staph infections. And then below that we have a bacillus type of bacteria, and it's basically just comparing a type of bacillus bacteria to a type of spirochete bacteria, or a bacterial cell with a spirillum shape. Bacteria can be killed with antibiotics. An antibiotic is a drug which interferes with the cellular processes of bacteria such that they die. And luckily, bacteria typically respond to antibiotics, and there are many different classes of antibiotics to choose from. There are various means by which antibiotics interfere with the cellular processes of a bacteria. They can prevent protein synthesis by binding to bacterial ribosomal RNA such that it can't be translated. They can destroy essential metabolic pathways. They can block DNA and RNA synthesis, which is going to effectively stop the bacteria from producing the proteins it needs for survival. And they can also affect bacterial cell walls, either breaking them down or preventing new cell walls from forming. However, there are several strains of bacteria which have become resistant to antibiotics and have been appearing in certain settings. So how do bacteria become resistant to antibiotics? Bacteria sometimes mutate and thus become resistant to antibiotics, though more commonly they can acquire a resistance gene from another bacterium, or rather we would say they obtain a plasmid. And a plasmid is a circular piece of DNA contained within a bacteria in addition to its double helix DNA. And bacteria can replicate these and then trade them off like trading cards in a process we call conjugation. Bacteria can become resistant to specific antibodies through several mechanisms. So there are different things the bacteria does after receiving this gene to become resistant to antibiotics. The bacterial membrane permeability can change, so the antibiotic can no longer enter. The antibiotic receptor protein on the bacterial plasma cell surface changes, so the antibiotic cannot attach. Their metabolism can be altered in such a way that the bacteria becomes capable of pumping out the bacteria or the antibiotic out of the cell. And they can also produce enzymes that destroy the particular antibiotic. So the best way for us to prevent any more bacterial antibiotic resistance is to make sure you take the full allotment of whatever prescription antibiotics you've been given, rather than stopping when you feel better. Because if you don't make completely sure that you've killed all the bacteria, you might have somehow brought about a mutated strain that's antibiotic resistant. Also, don't flush antibiotics down the toilet. I'm not sure why anyone would. And don't use antibacterial soap. And this is because essentially we're selecting for mutated bacteria that have developed a resistance to an antibiotic, such that we now have this big old new strain that can't be treated with antibiotics. One type of bacterial strain which is resistant to almost all types of antibiotics is methicillin-resistant Staphylococcus aureus, or MRSA. And typically, Staphylococcus aureus is a common bacterium on our skin, and this is just the non-resistant strain. However, infection with this strain can be serious or even life-threatening. Usually an infection must be initially caused by receiving a cut that the bacteria can get into. And MRSA is resistant to methicillin, as the name would suggest, amoxicillin, penicillin, oxacillin, and many other common antibiotics. However, there is one antibiotic which can be used to treat MRSA, and this is called vancomycin. And unfortunately, this is also one of the types of antibiotics that most people are allergic to. So I guess if you have an allergy to vancomycin, I'm not sure how well you would fare against MRSA. So now we're going to move on and talk about some different kinds of bacteria. And I think I've skipped around from what your book has laid out for you. But we're going to start with the plague, which is not just an ancient disease. And this is bubonic plague, which is a serious disease caused by the bacterium Yersinia pestis. And this type of bacteria is carried by fleas that live on rodents and is transmitted via the flea's bite. The first symptoms appear 2 to 5 days after being bitten, and these include high fever, rapid weak heartbeat, swollen lymph nodes, restlessness, delirium, and loss of coordination. And death can result in 3 to 5 days. So about as soon as you start seeing the symptoms, you may nearly be dead. Another type of bubonic plague is pneumonic plague, which infects the lungs, usually via inhalation. And the symptoms of this type of plague include sudden high fever, chills, rapid heart rate, severe headache, and coughing. And this form of the plague is highly contagious and can kill in 48 hours. There's also a third type of black plague, which is called septicemic plague, which in this type of plague, it indicates that the bacterium is actually in the bloodstream, and death can result without any symptoms appearing. So obviously, black plague is something that's pretty life-threatening if you actually have it. However, streptomycin is effective against most strains of plague, and we've pretty much effectively wiped out plague, except every once in a while someone might get sick of plague just out of the blue. And It's probably more common in third world countries than first world countries like the US. We've already talked about tuberculosis in the respiratory system module, but we're going to review it real quick again. And tuberculosis is a disease which usually settles in the lungs, and it's caused by a bacterium called Mycobacterium tuberculosis. It's transmitted via droplets in the air from the throat and lungs, and it usually results in or causes respiratory disease, and it can affect other organs other than the lungs. For example, it could cause a tubercular kidney. When tuberculosis enters the lungs, it may form a nodule of bacteria within the lungs, and when this happens, the infection is walled off and no further symptoms will develop. However, if some other respiratory or immune disease is present, this may activate the bacteria, leading to tuberculosis. And the symptoms of tuberculosis include coughing up blood, general weakness, weight loss, chest pains, fever, and night sweats. However, tuberculosis can be treated within 6 months with antibiotics. However, humans can actually carry this bacterium without any signs of infection, which makes it a much more prevalent disease than you would expect. And unfortunately, tuberculosis is quickly developing a resistance to antibiotics, so that's not good. Section 10.3, viruses can reproduce and kill, but they are not alive. Viruses are much smaller than bacteria and actually lack most of the characteristics of life, and one of these is that they can't actually carry out reproduction without the aid of their host. Viruses consist of only nucleic acid, and this can be either DNA or RNA, contained within a protein coat called a capsid. And viruses are actually cellular parasites, as I've sort of suggested, and they must contact a host cell, inject nucleic acid into the host, and take over its functioning. And they essentially turn this host cell into a virus-making factory. Viruses actually follow one of two types of reproductive cycles. There's the lytic cycle, where viruses cause the host cell to immediately become a viral factory, and then there's the lysogenic cycle, where the viral DNA is integrated into the host DNA, and then it can remain dormant for a while, and after a certain period of time, the host cell then begins to produce new viruses. Here are some pictures of viruses. In addition to the nucleic acid and capsid that make up a virus, some viruses have an envelope structure which they've stolen from the host cell, and this envelope structure is actually a small portion of the cell's plasma membrane, which the virus has wrapped itself in. And there are also these spike structures within the envelope. So here we have the adenovirus, which causes the cold. We have the herpes virus, which, you guessed it, causes herpes. And then we have this bacterial virus, which is a T4 phage, is what it's called. And this only attacks bacterial cells, despite being the scariest looking virus up here. And then we have the Ebola virus, which essentially just looks like a worm. Here's the difference between lytic virus phases and lysogenic virus phases. So in either of these cycles, the phage or the virus attaches to a receptor site on the bacterial wall. So this is a bacteriophage, and it penetrates the cell wall and inserts its DNA. The viral DNA is then transcribed and translated as if it belonged to the host cell. And here's when we go into our different cycles. So in the lytic cycle, we immediately start making virus particles. Many copies of the viral DNA and proteins are created. The protein coats are assembled and the viral DNA is packaged within them. And then the viral cells burst from the host. And this whole process of the host cell bursting is called lysis. And this releases new viral particles. However, in the lysogenic cycle, we incorporate the virus DNA into the DNA of the host, and the phage is replicated along with the bacterial DNA, and eventually we'll go back into a lytic cycle where where we begin making viral DNA and proteins, and then forming fully functional viruses which lyse from the cell. So essentially, in the lytic viral phase, the cell immediately starts pumping out viruses, whereas in the lysogenic cell, it could be a long time, years even, before the virus begins to, or rather, the virus is assembled and then lyses from the host cell. Most epidemics are actually caused by viruses, and here we have just a few viruses which have been epidemic in the past. We have the polio virus, which attacks the nervous system, and this was formerly a serious threat to infant health. Now we have a vaccine for it, and polio has been capable of causing total paralysis after just a few hours of being exposed to the virus. Then we have measles, which affects the lungs and is a threat to children. This is an incredibly contagious disease spread through person-to-person contact or inhalation of water droplets. Symptoms of measles include high fever, runny nose, cough, watery eyes, and some very characteristic white spots inside the mouth. And complications can arise from measles, which can lead to blindness, encephalitis, which is brain swelling, severe diarrhea, ear infections, pneumonia, or even subacute sclerosing panencephalitis, which is basically just another form of encephalitis. And of course, measles can lead to death also. There's hantavirus, which is a type of virus carried by mice, and this is contracted via inhalation of dust filled with dried and aerated mice urine and feces, and symptoms are flu-like, which makes it kind of difficult to diagnose at first. And then finally we have the West Nile virus, which is an avian virus contained within birds many times. This is carried by mosquitoes and can lead to inflammation of the brain and spinal cord. This inflammation and swelling of the brain is encephalitis, and I'm not sure exactly what they call it when the spinal cord swells. Ebola is a virus transmitted through direct contact, so one typically becomes infected with Ebola through contact with an infected individual's bodily fluids, such as blood or semen and other fluids. And these viruses cause hemorrhagic fever and are native to the rainforests of Africa. There are actually 4 types of Ebola. There is a non-lethal variety that actually lives in the western Pacific, and then there are 3 lethal strains which can kill from 50 to 90% of the people they infect. And this virus originated in chimps and gorillas, and we're really not sure at what point it jumped to humans. Symptoms of Ebola begin 2 to 21 days after contact, and there's several of these, which include sudden fever, intense weakness, severe headaches, muscle pain, and sore throat, followed by vomiting, diarrhea, and impaired kidney and liver function. And also the most characteristic symptoms of Ebola are internal and external bleeding, and I've heard it said that Ebola essentially jellifies your organs. And at the moment, we do not have an effective vaccine against the disease or any specific treatment, though I'm sure we will in the future. I hope, anyway. Despite how terrible Ebola sounds, the actual most dangerous virus is the flu virus, which travels the world. And the worst flu epidemic was the 1918 Spanish flu, which affected large portions of the world, and it's estimated that it may have killed in excess of 40 million people. And there are actually two types of flu. There is influenza B, which is the common cold, and influenza is actually where we get the word flu. We just take flu out of influenza. But anyway, influenza B is the common cold, and this is usually transmitted via inhalation, and the symptoms include respiratory distress, muscle aches, fever, chills, general lethargy, and severe headaches. And then influenza A is the strain which is responsible for the global pandemic. The code of this virus evolves quickly, making vaccines difficult to prepare, and unfortunately, this has the same symptoms as influenza B. There are also some other types of flu. There's H1N1 or swine flu, and H5N1 or avian flu. Section 10.4, AIDS and HIV attack the immune system. So AIDS, which is an acronym for Acquired Immune Deficiency Syndrome, formerly known as GRID, is actually a series of diverse symptoms associated with long-term infection by the human immunodeficiency virus or HIV. And the symptoms of HIV infection or the symptoms caused by HIV in AIDS include extreme weight loss, cancerous blotches on the skin, opportunistic infections, persistent fever with night sweats, swollen lymph nodes, and extreme fatigue. And despite the fact that AIDS seems like it is such a terrible virus, it can only survive for only approximately 20 minutes when in contact with drying air and oxygen, which is quite interesting actually. So this means that transmission of HIV occurs through body fluids. The live viruses can exist in semen, blood, vaginal secretions, saliva, and tears. However, transmission typically requires an open wound or other tear in the epithelial lining. And typically, AIDS is transmitted through unprotected intercourse with an infected individual or intravenous drug use, injection of drugs into the bloodstream. And these are the primary modes of transmission. Most viruses actually target a specific type of body cell, and the cell which HIV targets are the helper T cells, which they carry a protein called CD4 on their plasma membrane, which identifies them as helper T cells. HIV is what's known as a retrovirus. That means that the nucleotide contained within it is RNA, and this encodes its genetic instructions. However, when this RNA is injected into the host cell, it must be converted to DNA. And they do this through the use of a viral enzyme called reverse transcriptase, which reverse transcribes the RNA to DNA. Then this DNA is integrated or placed in the genome of the cell through the aid of a viral enzyme called integrase, which is responsible for both duplicating and inserting this, what we're calling cDNA, because it isn't technically a whole genome of DNA. That's what C means. And it inserts this into the host DNA. All retroviruses, not all retroviruses are lysogenic viruses, but AIDS is, and this explains why it may take 10 to 15 years for AIDS to develop. So here we need to know what viral load is, and this is the number of viral particles in the blood. And the viral load is higher after infection, and in the case of HIV, it drops as the CD4 T cells are infected. The number of T cells remains higher than the viral load during the first attack of HIV, but this viral load eventually becomes higher than the T cell count, and this is when the patient will begin to suffer from chronic infections. And this explains why HIV detection is difficult at times, because there's going to be an instance right after infection where you're not going to feel sick, and then maybe instances where you feel better. Those infected with HIV usually succumb to pneumocystis carinii pneumonia, tuberculosis, or Kaposi's sarcoma, which is a type of cancer caused by a virus. Here's the basic structure of HIV. It's 100 to 140 nanometers long, so it's about as big as the smallest type of bacteria. And in its envelope, it's made up of a phospholipid bilayer, as are all envelopes stolen from the host cell. And this phospholipid bilayer has the glycoproteins from the host cell embedded in it. And then we have this protein coat, also known as the capsid, if I didn't make that clear earlier. And then we have the single-stranded RNA within this protein coat, and the reverse transcriptase also. Here's how HIV reproduction works. So here at number 1, the virus attaches to the host cell at the CD4 receptor. So this protein embedded in the cell or the plasma membrane of the T cell is actually a type of receptor. The virus then injects its RNA into the cell, and with the aid of reverse transcriptase makes a complementary DNA or cDNA strand. And that's what I meant to say when I was talking about cDNA, sorry. The viral cDNA then makes a second strand of DNA, and this double-stranded viral DNA enters the nucleus and is then inserted into the host DNA, where it can remain dormant for many years as what is called as a provirus. So after several years have passed, the viral DNA can then be transcribed back into viral RNA and exploited into the cytoplasm. The viral DNA is then translated into new viral particles, and then the virus is assembled within the host and buds off a part of the host cell membrane to form the envelope. So now we have a fully functioning virus which has emerged from the host T cell. HIV treatment is still an uphill battle, and vaccines are difficult to make. So the current state-of-the-art treatment for AIDS is highly active antiretroviral therapy, or HAART, which involves the use of nucleotide analogs, and these are just drugs which resemble nucleotides, and protease inhibitors. These protease inhibitors work to block the enzyme protease, which is an enzyme also needed to produce new viral particles. So we're trying to inhibit the production of new viruses. And then the nucleotide analogs, such as AZT, prevent the creation of HIV proviruses in the infected cells. There isn't currently a vaccine for HIV because HIV simply mutates too quickly for vaccines to work. Many viral pathogens, including smallpox, polio, chickenpox, are all controlled by vaccines, and this is not the case with HIV. These vaccines, and we've covered this a little already, stimulate the immune system using an integral part of the viral coat, such as the portion that initiates contact with the T cell. And these aren't on the market, but they're being looked into and studied currently. Section 10.5 Other pathogens carry other dangers. So there are 3 other categories of pathogens which can attack humans, and these include certain types of fungi, protists, and prions. And we're just going to briefly cover each of these in probably one slide each. So be mostly concerned with the information related to bacteria and viruses. Fungi are eukaryotic organisms that play a major role in the decay process of dead organisms. And we went over fungi a little bit, I think all the way back in the first set of lecture slides. The diseases caused by fungi are typically more common in warm, moist environments, just because fungi favor these environments for growth. 2 types of fungal diseases are athlete's foot and yeast infections. Aspergillosis is a fungal infection caused by the fungi Aspergillus, and this disease leads to infection of the respiratory tract, which leads to asthmatic symptoms. And then there's zygomyosis, which is an infection of the blood vessels, which is predominantly found in patients with a compromised immune system. Fungal infections can be very difficult to treat because fungal cells are eukaryotic just like our own cells and are thus pretty similar actually. And we have antifungal drugs which can exploit the subtle differences. For example, fungi produce ergosterol, which is a lipid compound specific to their membrane that can be targeted for drug treatment. However, these antifungal medications can cause serious side effects such as chills, fever, and CNS disturbances, and this is related to the fact that antifungal medications are capable of killing our own body cells. So this is also why it's very difficult to get rid of fungi, like I said earlier. Protists are eukaryotic unicellular organisms, and they're actually responsible for a wide variety of diseases. For example, amoeba are a type of protist which can be contracted from contaminated water sources and are responsible for causing dysentery and sometimes meningitis or swelling of the meninges, which can be, well, both of these can be deadly. And then there's leishmaniasis, which is transmitted by the bite of a sandfly, and this leads to lesions on the skin or mucous membranes, and these lesions can be localized or distributed all over the skin. And then there's malaria, which is a disease I'm sure you've heard of, and this is caused by a Plasmodium protist carried by the Anopheles mosquito. Plasmodium matures in the red blood cells or erythrocytes of the host, and these erythrocyte cells are destroyed within the host. The symptoms include anemia, as we're killing all these red blood cells, fever, chills, nausea, and other flu-like symptoms. So the best way to prevent malaria is to control mosquitoes, and the best way to do this is just to dump any water that you see just randomly placed around. So if you see, like for instance, a kiddie pool which hasn't been dumped, it's probably a good idea to dump it, because this will serve as a very good breeding ground for mosquitoes. The last class of pathogen are prions, which are essentially just misshapen proteins, and they have even fewer characteristics of life than viruses. And this is because they are merely a protein with an odd conformation. However, what makes prion diseases so terrible is that prions are resistant to proteases and chemical degradation, which makes them almost impossible to destroy. And degradation is just the process by which we cause a protein, or in this case a protein particle, to lose its functional shape and then lose its in the case of prions, lose its ability to cause disease. When prions enter the brain, they can cause similar healthy proteins within the brain to unfold and then refold incorrectly, resulting in a chain reaction of destruction. So this unfolding and refolding actually works kind of like Alzheimer's does. And some types of diseases caused by prions are bovine spongiform encephalopathy, more commonly called mad cow disease, and the analogous disease to this is called Creutzfeldt-Jakob disease in humans, or Kuru, and there's also a disease called scabies in sheep, which I believe also causes some sort of sheep mange, essentially. And that's all there is for this chapter. And since we've covered how our immune system works, we're going to cover cancer in the next module and discuss how cancer develops and how the immune system may try to combat cancer.